Hi, I'm Jeff Corajod. I'm here with Jeff Deyoung, and our mentor was Dr. Li. We're from Hope College, and today we're going to talk a little bit about our project on Epifagus, which is also known as beechdrop. So basically we wanted to verify, in the past it's been assumed that beechdrops parasitize entirely on the roots of Fagus grandifolia, or the American beech tree. So we wanted to verify this using molecular studies, because the previous studies have all been they've just traced the host root all the way back to the tree, which can be time-consuming and painstaking process that also often has a lot of errors. So what we decided to do is we were going to go through and provide a molecular basis by we dug up the roots, as you can see here, this is the parasite and this is the root right here. And what we did was we took a small sample of the root and extracted the DNA from it, and the region we were particularly interested in was part of the chloroplast genome called rbcL. So we did a PCR to amplify the segment we were interested in, the rbcL gene, and then we used a big dye sequencing technique to determine the nucleotide sequences for the rbcL genes of all the samples we took. And I didn't mention, but all of our samples were from the area around Hope College, which is in West Michigan, as you can see on this map right here. So then we got the sequences, and Jeff's going to take it away from here, explaining what we did for analysis.
Alright, so after we gathered the sequences, which all came out to be around 1,300 base pairs long, which was come to be expected, we took the rbcL gene sequences of all the host plants that we thought had tissue connection with the parasite. We took those and we used POP to create a parsimony phylogenetic tree. As you can see here, 14 of our samples fit in with the clade of Fagus grandifolia, which was come to be expected. But then we had a red flag come up, and it was actually our sample from the host tissue. The sequence was very closely related to Acer saccharum, or sugar maple. This makes us ask more questions, like, is it possible that Epifagus does parasitize on plants other than Fagus grandifolia? And so with our future studies, we actually want to go through and collect more samples other than just 15 samples, and then also, instead of just using a dissecting microscope to see if there's tissue connection between Epifagus and the host plant, we actually want to cut slices out of the plant tissue and see if there's actual tissue connection by using stains and whatnot, and then making slides and looking under a microscope. And then so that's our future studies. Hopefully we can either prove the long believed assumption that Epifagus parasitizes only on Fagus grandifolia, or if we can discover that it actually can parasitize on further plants, which would be more beneficial for Epifagus. We'd like to acknowledge Dr. Li, his wife, and the Hope College Biology Department and NSF for funding, and thank you for your time.